All living things produce a waste product called hydrogen peroxide. Hydrogen peroxide is very nasty, it's a very unpleasant chemical, so it has to be broken down very quickly by living things. So living things have an enzyme called catalase, and catalase breaks down hydrogen peroxide to water and oxygen. As a source of hydrogen peroxide, we'll use celery. So what we'll do is we'll take a few stalks of celery and we'll chop it up. And celery will provide us with our enzyme catalase. Instead of celery, some people would use liver and various other materials. I'm using a chopping board to save the desk for safety reasons. And we'll transfer the chopped celery into a blender. Chopping up the celery breaks down some cell walls and helps to kind of blend it a little bit better. And blending it will release the enzyme from within the cells. Now, if we use a number of stalks of celery into the blender, so we add in some water, and blend for, we can only blend for a maximum of 6 seconds. So we'll blend for about 4 seconds. If you blend too long, the enzyme gets chopped up and gets destroyed, becomes denatured in that respect. So we'll blend for 4 seconds. We then have to filter it. We now have our enzyme prepared, and we have to filter it. To filter it, we use coffee filter paper. Coffee filter paper allows it to run through much quicker than lab filter paper. So pouring the filtrate in there, or the blend. And our filtrate comes through at the end. The filtrate then contains the enzyme. So we have to wait a moment for that to filter through. So having filtered it through, we transfer it to a beaker to allow us to measure it out. And I'm going to use a syringe to measure a certain amount of this into the graduated cylinders. Now, our celery, the filtrate is our celery, and the celery contains the enzyme catalase. We now have to transfer a certain amount of that into each of 4 graduated cylinders. We'll use this one with pH 4, pH 7, pH 10, pH 13. So if I transfer in 10 cubic centimeters of this, and into the second one the same again, 10 more. And into the third one the same again. And the same into the fourth one. And that gives us our enzyme solution. We then put in 10 of pH buffer. pH buffer guarantees or gives you a pH, a particular pH. This is pH buffer 4, so we use 10 cubic centimeters of that. And 10 cubic centimeters of pH buffer 7. And 10 cubic centimeters of pH buffer 10. And finally, 10 cubic centimeters of pH buffer 14. Now, the pH buffers guarantee us a pH of 4, 7, 10, and 13. The enzyme will break down hydrogen peroxide and produce oxygen. To get the oxygen to produce foam, we'll use a drop or two, two drops of washing up liquid into each container. So 2 drops of washing up liquid, 2 drops in here, 2 drops again, and 2 drops here. The substrate for the reaction is hydrogen peroxide. Hydrogen peroxide is a harmful material, so we'll use rubber gloves for that in case it splashes. It's a bleach and it will take the colour out of things. It's the same hydrogen peroxide that's used in bleaching hair and things like that. Hydrogen peroxide has to be kept in the dark because it breaks down in light. So we'll transfer some to a beaker. And with the hydrogen peroxide, we'll pour some 5 cubic centimetres into a small graduated cylinder. So just 5 cubic centimetres of this. Five into the second one. Five cubic centimetres of hydrogen peroxide again. And finally, 5 more into this graduated cylinder. Now, we have to bring all of these to the same temperature. They may be at different temperatures. So to bring them to a suitable temperature, catalase from celery would work fairly well at 25 degrees. So we'll transfer them all for 10 minutes to a water bath at 25 degrees centigrade. And we leave them here for 10 minutes to reach a constant temperature of 25 degrees centigrade. After 10 minutes, everything has reached 25 degrees centigrade. So we take them out of the water bath. And we have the enzyme in this large graduated cylinder. We have the substrate hydrogen peroxide here, so we mix the two together. We pour the hydroxide down the side. Like so. At pH 7, there's not much foam. The foam, we'll see it in the next one, there's not much foam here because at pH 7, the enzyme is denatured. The enzyme is not working properly. In the second one, and we add the hydrogen peroxide down the side, and straight away we see bubbles. And tiny bubbles are formed down here. The bubbles react. The bubbles are oxygen. They react with the washing up liquid to produce foam. We wait 2 minutes and we'll record the level of foam after 2 minutes. In the next one, we add the hydrogen peroxide down the side. And again, foam is produced. And again, we'll wait 2 minutes to record the level of the foam. And the final one, pH 13. We add the hydroxide down the side. And in this case, again, no foam is produced. That indicates that the enzyme isn't working. The enzyme has been denatured by the unsuitable pH. We wait 2 minutes to record the level of foam in each of these containers. After 2 minutes, at pH 4, we have no foam. The enzyme was denatured, the enzyme didn't work, no foam was produced. At pH 7, we have a certain amount of foam. We have to subtract the volume of liquid from the volume of foam. So therefore, when we work it out, we get 64 cubic centimetres of foam. At pH 10, subtracting out the volume of liquid from the foam, we get 78 cubic centimetres of foam. So the enzyme has worked best here. This is the optimum pH. And at pH 13, no foam, the enzyme again was denatured. So we've shown that the enzyme worked best at pH 10, it worked reasonably well at pH 7, but didn't work at pHs 4 and 13.